Hello friends, in the series of IITPL, I am Payal Pridarshini, PGT Biotechnology, Kendriya Vidyalaya number 3, Delhi Cant. Today, we will learn about genome organization and chromosome. Before going in detail in this topic, genome organization and chromosome, let us understand the meaning of genome and organization. Genome means the complete set of chromosome or genetic material present in a cell or organism. That means whatever DNA is present in our cells or in any living organism, that all together is called genome. And organization means arrangement of anything for certain purpose. That means if anything is arranged in a particular manner for a particular purpose, that is called organization. So, in the same way, our DNA, our genome are arranged in a particular manner to fit into the cell or nucleus. So, here comes a question, why genome organization is required? Why it is needed? Why genomes needs to be packed inside the cells or nucleus? And the answer is the length of DNA with respect to size of cells and nucleus. You can see over here, this is a DNA and this one is a small cell or a nucleus. These long DNA needs to be packed inside a small cell or nucleus. This is a very tedious task for the DNA to get packed inside a small cell or nucleus. Say suppose you have been given anything whose size is very much larger than that of the box. So, how could you fit that large object in that small box? For that, there must be some organization of that object to fit into the small box. In the same way, this DNA packed itself with the help of certain proteins to pack inside a small cell or nucleus. In this series, today we will learn about genome organization of virus. As we know, virus is intermediate of living and non-living organism. When it is present inside a cell, it becomes a living organism. So, how the genomes are there in the virus? What are the different features of the genome in the virus? That we will learn in this series. Next, we will learn about genome organization in bacteria. As we know, bacteria is considered as prokaryotic cell. Pro means primitive, karyot means nucleus. So, bacteria is considered as prokaryote because they do not have a nuclear membrane to enclose the genetic material. So, how the genome is present inside the bacteria, that we will learn in this series. Next to that, we will learn about genome organization in eukaryotes. Eukaryotes, eu means true, karyot means nucleus. So, eukaryotes are those cells, those living organisms which have true nucleus, which have nuclear membrane to enclose the genetic material, that is DNA or chromosome. In this topic, we will learn how the genome of the eukaryotes packed themselves inside the cell, inside the nucleus with the help of certain protein. Next to that, we will learn about structure of chromosome. What are the different parts of the chromosome? We will learn in this series. Next to this, we will learn about types of chromosome. Depending on the position of centromere in the chromosome, we will learn about types of different types chromosomes. Next to this, we will learn about the function of chromosome. What are the different functions performed by the chromosome? and after the completion of this series, at the end I will give you the answers of the question which I asked in my last session on RNA, and we will ask you some question of this series. So, let us start with the genome organization in virus. If we talk about the features of viral genome, the genome can be single stranded DNA, the genome can be double stranded DNA, the genome can be single stranded RNA or double stranded RNA. So, all the 4 types of nucleic acids are present in case of virus only, and the genome of virus may be a linear molecule or circular. In continuation with this, some more features are there of viral genome. The viral genome, either it will be a one molecule or it may be a segmented genome, that means they may be fragmentized. The virus which contain RNA as a genetic material are considered as retrovirus. The genome size of the virus may range from 2 kilo base pair to 2 million base pairs. And the most important thing about the viral genome organization is there is no any specific pattern of organization in the virus. The genome are present without any help of any protein inside the virus. As we know, virus is having 2 parts. One is the proteinous part which form the core or capsid, and another is the nucleic acid. So, there is no any organization, there is no any particular way to pack the DNA or RNA inside the virus. Now, one more question which comes in our mind is, how do bacteria lacking a nucleus organize and pack their genome into the cells? That means, if we compare the length of the DNA and the size of the bacterial cell, the length of the DNA is 1000 times larger than that of the volume of the cell of bacteria. So, how could 1000 times larger DNA can be incorporated in a small bacterial cell? Before discussing the packaging of the genome inside the bacterial cell, let us know about the features of bacterial genome. Most genome of bacterial cells are single molecule. They are not fragmentized as that of the virus. They are circular in nature. The DNA which is present in bacterial cell are not linear, but they are circular. The DNA is double stranded. Double stranded means, according to Watson and Crick, DNA is double stranded, they have 2 polypeptide chain. Like that, the same DNA is present in the bacteria also. But in case of virus, the virus may have single stranded DNA, double stranded DNA, single stranded RNA and double stranded RNA. But in case of bacteria, only double stranded DNA is present, and some additional circular double stranded DNA is also present in the bacteria, which is called plasmid. The bacterial genome, the bacterial DNA is organized in a structure which is called nucleoid, and the size of bacterial chromosome ranges from 0.6 million base pair to over 10 million base pair. Now, let us see how the bacterial genome packed itself, organized itself, incorporate itself in a bacterial cell. So, there are 2 levels of genome organization in bacteria. One is loop formation and second one is supercoiling. First of all, the bacterial cell will form the loop with the help of certain protein, and after the formation of loop there will be supercoiling in the loop to increase the size of the DNA so that they can easily be packed inside a bacterial cell. Now let us see how the loops are formed inside the bacterial cell. So this is a bacterial genome which is double stranded and circular. As I told you earlier, these bacterial genome has to be folded 1000 times so that it can be incorporated into the bacterial cell. So here you can see, let us suppose this is a bacterial genome which is very much larger in size, and if we talk about the bacterial cell, this is a bacterial cell. If we compare the size of bacterial cell and the bacterial genome, you can see the size of bacterial genome is much more larger than that of the size of the bacteria. So how this large size bacterial genome will incorporate in a small bacterial cell? So these bacterial cell, with the help of proteins, will make the fold. There are mainly two types of protein. The one is IHF, that is integrated host factor, and second one is DNA binding proteins. These two proteins will help the bacterial genome to incorporate in a bacterial cell which is very small in volume. With the help of these two proteins, these bacterial genome will form the loop. Let us see how they will form the loops. This is the way how the bacterial genome form the loops. These are the loops which are formed by certain proteins which I mentioned earlier. The proteins are IHF and DNA binding proteins. These proteins help the bacterial genome to form the protein, and all these proteins are present in the center of this particular structure which is called nucleoid. This particular structure formed by the bacterial genome with the help of proteins is called nucleides. Now these loops of the bacterial genome again form the supercoilings. You can see in this slide, these loops will again get coiled, supercoiled to reduce the size. If you see the difference in their sizes, so these loops will form a negative supercoilings like this to reduce the size. Like this, every loop get supercoiled and negatively supercoiled, and because of this negative supercoiling, the size of the DNA get reduced, and because of the decrease in the size of the DNA, this DNA of bacteria can easily get incorporated into the bacterial cell. Now let us know more about the supercoilings. Supercoilings is the rotation of DNA molecule which may be positive supercoiling or negative supercoiling. Consider this as bacterial genome which is in a relaxed state in a circular manner. If we see this DNA is right-handed, and this right-handed DNA, if again moved in right-handed position, so it can form a supercoilings which is called like this. You can see here this form the supercoilings, and these supercoilings are called positive supercoilings because the rotation is over and rotation is right handed as that of the coiling of the DNA, and if it is rotated opposite to the direction of DNA coiling, it is called negative supercoiling, and negative supercoiling help in the packaging of the DNA in the bacterial cell. So, till now we learned about viral genome and bacterial genome. Bacterial genome organizes itself with the help of two different processes. One is loop formation with the help of integrated host factor and DNA binding proteins, and again those loops get supercoiled to reduce the size, and in that supercoiling two types of enzymes are involved. One is DNA gyrase, which is also called DNA topoisomerase 2, which introduces negative supercoilings using energy from ATP, and it also relaxes positive supercoils when they occur, and DNA topoisomerase 1 relaxes negative supercoils. So, these are the two enzymes which help in the supercoilings of the bacterial genome. Now, here are some more information about the supercoiling. The chromosomal DNA in bacteria is negatively supercoiled, which I told you earlier. Negative supercoil in bacteria help the DNA to get packed inside the small bacterial cell, and this negative supercoiling is helped by an enzyme called topoisomerase. Negative supercoilings has two major effects. One, it helps in the compaction of the chromosome. Because of the supercoilings only, the bacterial DNA reduces its size to get incorporated into the bacterial cell, and secondly, creates tension that may be released by DNA strand separation. During the DNA replication, both the strands are being separated because of the tension created by negative supercoilings. So, this is what the genome organization in bacteria. Let us recall at once. If we compare the size of the bacterial cell and the genome present in the bacteria, the size of the DNA is 1000 times more than that of the size of the bacterial cell. So, the bacterial genome has to be organized in such a way that it can be incorporated into the bacterial cell. For that, bacterial genome forms the loops with the help of protein called IHF, that is integrated host proteins, and DNA binding proteins. After the formation of loops, the loops get supercoiled, and that supercoiling is negative supercoilings which help further packing of DNA length, and this is how the bacterial genome get incorporated into the bacterial cell. Now, let us know about the genome organization in eukaryotes. Do you know what are eukaryotes? Yes, eu means true and karyotes means nucleus. So, all those living organisms which have true nucleus, which have nuclear membrane to enclose the genetic material, are considered as eukaryotes. You, me, and all the living organisms, all the animals and plants are considered as eukaryotes because we all are having true nucleus That means our genetic material, our DNA, our chromosomes are enclosed in a membrane which is called nuclear membrane. Now let us know about how the genome of eukaryotic cells are organized inside the nucleus of the eukaryotic cell. So if we talk about eukaryotic genome, genome is double stranded DNA. Our genome are double stranded in nature, and genome arranged in a linear structure called chromosomes. In case of all eukaryotic living organism, the genome is linear, it is not circular as that of the bacterial cell. So if we talk about genome organization in eukaryotic cell, while comparing the size of the DNA and the size of the nucleus, the size of the DNA of the eukaryotic cell, especially in human cells, it is 14,000 times larger than that of the size of the nucleus. So we cannot imagine how such a long DNA can be incorporated into a small nucleus of eukaryotic cell. But anyhow, the DNA packed itself in a particular manner to get inside a small nucleus. So there are some labels of DNA packaging of the eukaryotic cell in the nucleus. The first label is formation of nucleosome. Nucleosome consists of histone proteins and a DNA which is wrapped over the histone protein. Second label is formation of chromatin fiber. The nucleosome get compact to form chromatin fiber, and our genetic material in various phases of the cell cycle are appear in fibers structure. The third level of DNA packaging is loop domain formation. These chromatin fibers further get condensed to form loop domain. Next to the formation of loop domain, there is formation of heterochromatin. That means these loop domain get further condensed to form heterochromatin, and heterochromatin is highly condensed part of the chromosome. And finally, the metaphase chromosome is being formed after the further condensation of chromatin and heterochromatins. Now let us move in detail of all these steps. The proteins which involve in the DNA packaging, the one is histone proteins. There are 5 types of histone protein: H1, H2A, H2B, H3 and H4. These are the 5 types of histone proteins which involve in packaging of the DNA. And second number of protein that are called non-histone protein, they also directly or indirectly help in the packaging of the DNA. And non-histone protein includes scaffold protein, DNA polymerase, heterochromatin protein 1 and polycom, and so many different proteins are also there. So these are the 2 proteins which involve in the DNA packaging, which helps the DNA to reduce its size. One is histone protein. There are 5 types of histone protein: H1, H2A, H2B, H3 and H4. And non-histone protein are scaffold protein, DNA polymerase, heterochromatin protein 1 and polycom. Now the question which comes in our mind is, why DNA wrapped on histone? Let us know how these proteins help the DNA to reduce its size. So, these are the 5 types of histone protein: H3, H4, H1 and H2A, H2B. So, if suppose this is a H3 protein, this one is H4 protein, this one is H2B protein, this one is H2A protein and this one is H1 protein. So, these are the 5 types of histone proteins which help in the packaging of DNA. 2 molecules each of H3, H4, H2A and H3, they will combine together to form histone octamer. Let us know how this histone protein combines to form histone octamer. H3, H4, H2A and H2B will combine to form tetramer, and again the another molecule, second molecule of the same protein will combine to form histone octamer. You can see 2 molecule each of H3, H4, H2A, H2B combines together to form histone octamer. Let us consider this as histone octamer having 2 molecule each of H3, H4, H2A and H2B. And these histone protein are positively charged because of arginine and lysine. As we know, proteins are polypeptide chain of amino acid. They are made up of amino acid, and arginine and lysine have positive charge, and because of the presence of arginine and lysine in the histone protein, the histone proteins are positively charged. And if we talk about DNA, DNA is negatively charged. Do you know why DNA is negatively charged? Yes, you are right, DNA is negatively charged because of the phosphate group present in the DNA. And this histone is positively charged, and let us suppose this is the DNA, this DNA is negatively charged. Because of the opposite charge, this histone will help the DNA to get incorporated, to get wrapped on this. Now, let us see how the DNA get wrapped over the nucleosome, histone octamer. This is the way how a DNA get wrapped over histone octamer. One complete turn, 1.76 turn is there. So, the DNA get wrapped over the histone octamer, and this complete structure which includes histone octamer and this DNA is called nucleosome. This particular structure is called nucleosome, and in the same way so many histone octamers is being synthesized which help the DNA to get wrapped over them, and the size of the DNA get reduced, and because of this histone proteins, the DNA get easily packed inside the nucleus. So, you can see many nucleosome inside the nucleus of the eukaryotic cell, So, this way various nucleosome you can find inside the nucleus of the eukaryotic cell. This protein is called H1 protein, and the DNA between two nucleosome is called linker DNA. This H1 protein will help for the further packing of the nucleosome to form the next level of DNA packaging, that is 30 nanometer condensed chromatin fiber. So, you can see in the slide, this is how the histone octamer is being synthesized and DNA is being wrapped over here. The DNA in between the two nucleosome is called linker DNA, and in the nucleosome, in the outer part, there is H1 histone protein which help in further packing of nucleosomes to form 30 nanometer chromatin fiber. This nucleosome model was given by R.D. Kornberg, and the length of this nucleosome is approximately 11 nanometer. So, if we see the arrangement of this nucleosome in the nucleus, you can see the beads like structure on the DNA. You can see the beads like structure in the chromosome because of the presence of nucleosome. These beads like structure we can consider them as nucleosome, and because of the H1 protein they get further coiled to form 30 nanometer chromatin fiber. So, you can see the length of the DNA is very much larger still. So, it get coiled like this to form 30 nanometer chromatin fiber, that is the second level of DNA package. Next to this chromatin fiber, with the help of some non-histone protein which is called scaffold protein, get more condensed to form 300 nanometer fiber. You can see in the slide, these are the 30 nanometer chromatin fiber with the help of non-histone protein get further coiled to form loop domain and reduces the size of the DNA, and the size of this particular structure is 300 nanometer. Next to this, the 300 nanometer fiber get more condensed to form heterochromatin of 700 nanometer. Level of DNA compaction changes throughout the cell cycle, most compact during M phase and least compact during S phase. M phase is the division phase of the cell and S phase is synthesis phase of the DNA. In S phase, the DNA get replicated. So, DNA is very compact during the division phase and they are less compact during the synthesis phase. There are two types of chromatin related to the levels of gene expression. One is euchromatin and second one is heterochromatin. Euchromatin is that area of chromosome that stain lightly. If we color, if it provides any dye in a stain to the chromosome, that area of the chromosome which get stained light are considered as euchromatin, and heterochromatin are those area of chromosome which stains darkly. So, this is how we can differentiate the euchromatin and heterochromatin part of the chromosome, which is mainly based on staining with certain dye. Euchromatin exhibits normal pattern of condensation and relaxation during the cell cycle. As euchromatin is less condensed, so it can undergo the process of condensation and relaxation during the cell cycle, while heterochromatin at that region of chromosome which is highly condensed and do not go under condensation and relaxation because they are highly condensed, and highly condensed helps the DNA to reduce its size. You can see in this slide euchromatin is uncoiled and active, whereas heterochromatin remains condensed and is inactive. Euchromatin is considered as the active part of the chromosome or functional part of the chromosome which undergo the process of replication and transcription to synthesize the RNA, while heterochromatin is highly condensed part of the chromosome which do not undergo replication and transcription. And the last stage of packaging of DNA in eukaryotic cell is formation of chromosome, which we can see in metaphase of cell cycle. This is how the chromosome is being synthesized or formed because of the packaging of the DNA. The length, the width of the chromosome is approximately 1400 nanometer. So, this is how the chromosome formed inside the nucleus of the eukaryotic cell with the help of DNA packaging and certain proteins. Let us recall the packaging of the DNA inside the nucleus of the eukaryotic cell. This you can see the DNA, and according to Watson and Crick, the width of this DNA is 2 nanometer. This DNA will first of all get wrapped on the histone octamer to form the nucleosome, and the length of this nucleosome is 11 nanometer. These nucleosome will further condense to form 30 nanometer chromatin fiber, and these 30 nanometer chromatin fiber with the help of certain scaffold protein further forms a loop of 30 nanometer, and these 300 nanometer loop get again condensed to form 700 nanometer heterochromatin, and these also get condensed to form 1400 nanometer wide chromosome. So, this is how a long DNA get packed inside a small nucleus of eukaryotic cell. Now, let us move towards the chromosome. Chromosome derived from a Greek word in which chroma means color and soma means body. So, chromosome can be stained by certain dye to provide the color, and because of the color pattern given by the chromosome, they have been given the name chromosome. Chromosomes are thread-like structure located inside the nucleus of the eukaryotic cell. If you can see the chromosome with the help of electron microscope, you can find thread-like structure inside the nucleus of the eukaryotic cell, and those thread-like structure is considered as chromosome. Now let us know about the different parts of the chromosome. The first is pellicle and matrix, second one is chromonemata, third one is chromomeres, fourth one is centromeres, fifth one is secondary constrictions, sixth one is satellite bodies, and seventh one is telomeres. Now let us know about all these parts of the chromosome. First about pellicle and matrix. This outer layer, this outer membrane of the chromosome is considered as pellicle, and this membrane encloses a jelly-like structure which is called matrix. So, pellicle bound the membrane to give the structure which appears during the M phase of the cell cycle. Next to this thread-like structure in the chromosome is considered as chromonemata. Chromonemata is two identical spirally coiled thread, and on this chromonemata you can find this bead-like structure, and this bead-like structure is called chromomeres. Next to this, the region in the chromosome which is less condensed is called centromere or primary constriction. At the primary constriction or centromere, both the arms of the chromosomes attached. Next is secondary constriction. Secondary constrictions are those regions of the chromosome where the nucleolus get assembled. Beyond these secondary constrictions, some fragments are there which is called satellite bodies, and at the tip of these chromosomes telomeres are present, which has a unique property that it prevents the end of the chromosomes from sticking to each other or one another. So, these are the different parts of the chromosome. Let us move towards the types of the chromosome. There are mainly 6 types of chromosome depending on the position of centromere, that is primary constriction, which joins the arms of the chromosome, and these types are metacentric, submetacentric, acrocentric, telocentric, acentric, and dicentric and polycentric. Metacentric is that chromosome in which centromere is present near about in the middle of the chromosome. You can see in the slide, this is the position of centromere which is exactly at the middle of the arms of the chromosome, and both the arms are equal in size. That is why they give V-shaped chromosomes. So, we can recognize the metacentric chromosome as V-shaped. Next to metacentric, submetacentric chromosome is there. The centromere is not present in the center of chromosome. Because of that, both the arms are unequal in size, and they gives L shape to the submetacentric chromosome. Acrocentric, the centromeres are present at the one end of the chromosome. The one arm of the chromosome is little bit smaller than that of the another arm, and because of that they appears J in shape. Next is telocentric. The centromere is present at the proximal ends, terminal ends of the chromosome, which gives I shape to the chromosome, and this is called telocentric. Acentric are those chromosomes which are lacking centromeres, and dicentric and polycentric are those chromosomes which have more than one centromere. If they have two centromere in them, they are considered as dicentric, and if more than two centromeres are there in chromosome, they are considered as polycentric. Now let us know something about the function of chromosome. So the first important function of chromosome is they contains hereditary information in the form of genes and act as hereditary vehicle. As we know, chromosome is having DNA, and DNA is considered as genetic material because it transfers the genetic information, characters, traits from one generation to another. So those DNA is being transferred from one generation to another with the help of chromosome. Second most important function of chromosome is they control division, growth, metabolism and differentiation of cell. The chromosome will help in the cell division, in the growth of the cell, various metabolic reactions of the cell and differentiation of cells. The next function of chromosome is it determines the sex of individual. As we know, there are two types of sex chromosome present in human body. One is X chromosome and another is Y chromosome. So this sex chromosome help in the determination of sex of the individual. Next to this, another important function of chromosome, the crossing over and aberration of chromosome introduce variation in population. As we know, no individuals are very much identical to each other, and this is because of the crossing over and changes in the shape, size in the chromosome, and because of that there is variation in the population. So let us summarize all the topic which we learned today. Why DNA packaging is required? DNA packaging is required to reduces the size of the DNA, because if we compare the size of the DNA and the size of the cells or nucleus, the length of the DNA is much more than that of the volume of the cells or nucleus. So packaging is very much important to reduce the size of the DNA so that they can easily incorporated inside the cells or nucleus. Then we discussed about viral genome. As virus have all the 4 types of nucleic acid, that is single stranded DNA, double stranded DNA, single stranded RNA and double stranded RNA. So all these 4 types of nucleic acids are there in the virus. There is no any particular way of organization of viral genome in the virus. They do not need any protein to pack their genome inside the core or capsid of the virus. The genome of the virus may be a single molecule or fragmentized. Next to this, we discussed about genome organization in bacteria. As we know, bacteria lacks the nucleus, but comparing the length of the DNA and size of the bacteria, the length of the DNA of bacterial genome is 1000 times more than that of the size of the bacterial cell. So, in case of bacteria also, the DNA needs to get reduced its size. For this purpose, the bacteria requires two different types of protein, that is integrated host factor protein and DNA binding proteins. These two proteins help the bacterial genome to reduce its size by forming the loop, and again those loops get supercoiled, negatively supercoiled to reduce its size further. Next to that, we also learn about DNA packaging in the eukaryotes. In case of eukaryotes, the DNA, with the help of certain proteins like histone proteins and non-histone proteins, reduces its size to get incorporated inside the nucleus of eukaryotic cell. Then we discussed about the structure of chromosome, types of chromosome and function of chromosome. Now, you need to test yourself, answer all these questions. What are nucleoids? What are the differences between positive and negative supercoilings? Why DNA packaging is needed? What are the different proteins involved in nucleosome model? And what are the different types of chromosome depending on the centromere position? In my last session, I asked some question, what are the differences between DNA and RNA? So, these are the differences. The another question which I asked in my last session, you need to write the RNA sequence of the given DNA. In spite of writing T, you have to write U, because RNA is not having thymine nucleotide, they have uracil nucleotide. So, answer is this. And the last question which I asked in my last session was the function of following RNA, messenger RNA, transfer RNA and ribosomal RNA. The answer is messenger RNA codes for amino acid, transfer RNA bring the amino acid during protein synthesis, and ribosomal RNA help in the synthesis of ribosome. Here ends the today's topic, that is genome organization and chromosome. In the next session, we will learn about the search of genetic material, DNA application and all. Thank you very much.